cold. I go outside.
All right, we're going to go ahead and get started, everyone. So we've got about an hour and 40 minutes left. This is going to be a discussion that Bob and myself, Sandy, will moderate. But we're wrapping up. We're almost there. I'm going to share a couple of slides here. We had a session at 2 that we were going to talk about opportunities to utilize the standard for improving communication of device performance. So I want to start off the discussion with that, and then we're going to come back to some of the things from this morning about skin pigment quantification and try and develop some more consensus and than other issues. Give me just 1 second here.
Mike, what do you mean communication? In what sense? Communication to end users or communication?
Good question. So I put up here, I was trying to articulate this and arguably didn't do a good job of it, but with regard to communication, there's a couple different buckets where there seem to be communication problems based on either what we've been hearing, what people have proposed in prior meetings, and here's my best attempt to do this. One is communication of the expected performance, the intended use, durability for somebody who is procuring devices, like a Ministry of Health, an aid agency. What devices should I be buying for the setting that I'm buying the devices for? There seems to be confusion about that. We saw during COVID and even before COVID, many devices being bought at the health system level that should not have been bought. But it was confusing and hard to navigate for some of the people making those procurement decisions. So that's one area of communication that perhaps can be better addressed. Another was communication of expected performance to the end user. I got this device donated to me. What do I expect it to be? How should I expect it to perform? There's confusion around that, and some of that's related to number one, of course. And then finally, at the bedside, what is the device communicating to me on this measurement in this patient? How competent is it in that? And how is it communicating to me, whether as a clinician or as an end user in home care setting, for example, how much do I trust that? Can the device do a better job of communicating certainty, uncertainty? Can it do a better job of communicating signal quality? And so I think that was sort of how I had broken that down. The other piece of this, well, that's somewhat related to this kind of the communication of expected performance, intended use, is this issue of categorization. Glory had brought this up in the prior presentation about intended use and durability and the flood of pulse oximeters on the market. I don't know that I have the experience or perspective to make the statement with confidence, but it seems to be a pretty unique situation where a medical device has come onto the market in such volume and become a household product in some ways, but there's huge variability in the performance, and it's not easy to figure out which ones are good and which ones aren't, especially for these lower cost fingertip devices in particular, is where that was seen. We were seeing donations of tens, hundreds of thousands of devices, and 3 months later, none of them worked because they weren't context appropriate. They just don't last in that environment. So the issue of performance also and wellness devices, these are some of the product codes from the FDA categorizing. I don't want to speak to describe this, but this issue of devices that are intended to be for quote wellness that often are being used, bought perhaps inadvertently or intentionally, but maybe shouldn't be. Should there be a higher bar for those? Should those be on the device on the market when in fact the only thing that they're cleared to be doing is measuring a heart rate, but they're still reporting another number, and not everybody knows that may be the case. And so wanted to open this up for discussion. Those are some of the issues that we're commonly hearing. Is there a way that this can be improved upon by using the standard or changes to the extent that that's even possible with how we approach pulse oximeters as a quote wellness device. Paul, I see your hand, and then we'll go to James Lee, who's been online and waiting patiently.
Oh, well, yeah, I'll cut it short. I'm not aware that the PGJ Oximeter Wellness or the OCH oximeter or infrared sporting aviation, that either one of those are required to have hypoxia accuracy testing.
I believe that's correct.
So that's a big issue.
It is, especially for aviation. I mean, it'd be nice to know that you're not going to black out at the wheel.
Sandy, I see your hand, and then James Lee, if you're on still, would appreciate any context and comments from you. Sandy, over to you.
I just wanted to make a few quick points. We, in the standards committee, wrestle with the intended use, the context of use, the use environment all the time, and try to understand whether there is any, if you will, demarcations between the different flavors of oximeters. And to date, we haven't really been successful trying to figure that out. So, for example, you say, well, home use devices, maybe they can be cheaper, but the current approach from a standards perspective is home use is a much more, I was going to say, crappy environment where things get beat up. And do you want any less accuracy in a home use device than you do in a professional device? So I'm just throwing that out there for discussion and some of the kinds of questions that you have to wrestle with.
Thanks, Sandy. Dr. Lee, over to you. Thanks for waiting patiently. Appreciate you joining.
Sure. Hopefully I can provide some, background here. So I think the comments regarding product codes, just for clarity on this slide, these are all the product codes in a particular regulation. And that's and I'm sorry if this is a little bit wonky, but that's CFR 870.2700. And so those are all the basically the basic technology functional type of bucketing that the agency did for now several decades. And these devices are all light-based oximeters. There are only a handful of them that you would be familiar with in the context of oximetry and getting a SPO2 number. And just to be clear, DQA is the product code which most of these fingertip pulse oximeters would be sitting. Those are prescription use types of devices for medical purposes, and the commensurate standards and testing would apply. So if you go to the FDA database, you look at a regulation or a particular product code, that product code regulation would have a definition, which regulation and any pertinent standards that are used. And so if you look at DQA, you'll see all the biocompatibility standards, which are like ISO 10993, and you'll see the one that the committee here is discussing, the 80601.2.61, right? And so those are the ones where a company comes in, they will declare that they conform to these standards because they're recommended by the agency. And those standards, when you follow them and you adhere to them, you would require the desaturation studies. And I think I heard the question about what about these other devices that are wellness? They don't necessarily have to use the same type of clinical study, but it doesn't mean that these companies did not. And so that's where I think, and part of me, I'm not a worldwide regulator, right? We just look at US FDA, but I know for CE marks, many of the CE marks would still utilize the ISO standard as a benchmark. And so that's why the standard is so important. And it provides, relatively level playing field for basically pressure testing the device, evaluating, validating the performance of the device. And that's why your standard is so important. There is a question, though, I think I heard about, whether or not the utility of sports and aviation or wellness devices, if they are not compelled to do review. Now, meaning the issue here, though, is that these devices are still devices. But the agency basically, because of the low risk of the use, like if you're using it for exercising and such, recognize that use as lower risk, and thus the FDA does not need to see the test and validation prior to a company selling it. It does not mean that the company gets off the hook for truth in labeling. So you would still, whatever labeling the company has provided about how they validated their technology, that's still truth in labeling, and that those any claims there would still need to be, they would have to be truthful about that information. So part of the issue, I think, if you need clarity about a particular technology, when it does not appear that it has the FDA 510K, is first look at that labeling. Is the device CE marked? Is the device have declarations about accuracy? And anything that they have, they would, should be truthful. I'm not sure if that entirely helps your discussion, but just for context, the agency looks at a top-down approach when we look at a medical device. First and foremost, they look at the intended use. And if the intended use is not for, let's say, medical care, that, relegates it to the other product codes that aren't medical devices like TGJ or OCH. Hope that's helpful.
Thanks very much. Other comments?
My biggest concern is if a device puts the words Pulse Oximeter on it, that's a claim, it seems to me. And people will use it for that. And we've looked at a lot of over-the-counter devices that do not measure pulse oximetry. But they still say they're a pulse oximeter.
Thanks, Paul. Julian, comment to you.
Yes, thank you. So I think of how non-invasive blood pressure monitors are marketed and sold. They are medical devices. They meet appropriate existing standards, and they're available for sale without a prescription to the population. It seems to me that they're used appropriately. So why can't we treat pulse oximeters the same way? So treat them as medical devices available without a prescription, available to the population, and then they'll have the benefit of better instruments, and we can continue with education. I mean, I've had plenty of patients. What's that? They're cleared.
They are cleared.
Yeah, they're cleared. They're cleared. They meet standards. They're cleared. They're medical devices, and they're widely available on Amazon. It seems like a system that works quite well.
Correct.
I don't know why we don't mimic that for pulse oximetry.
David,
Before I go to your comment, Dr. Lee, do you want to make any response to Julian or Paul's comments?
Sure. Thank you for that. And thank you, Dr. Goldman. You're correct. Blood pressure cuffs are class 1 medical devices. I think here is that, and it might, part of that, it has to do with the marketplace. There are a lot of companies that make devices or that make technology that they clearly do not want it for medical purposes, like sports. I won't name companies, but there are many of them that look at oximetry, look at heart rate, look at other functions. And so I think that's a distinction, but your point's well taken. If a company wants medical claims, then they should be, they should have some validation. And classification for class 1 or class 2, there is a possibility that you could have, certain devices that are medical that are lower risk, but that's not happened yet. One of the things I would say, though, is when we look at over the counter, particularly regarding kind of information that a pulse oximeter would give, like saturation, being over the counter means that you don't need a learned intermediary, that the person on their own, they could act as their own learned person, looking at the number and making, be situationally aware of their current health status. And I think that's one of the things that the agency also would be sensitive to. But good points. Thank you.
Could I just ask for clarification? So it's my understanding that manufacturers are not permitted to sell pulse oximeters over the counter if they are treated as medical devices and cleared, that they don't have that option, and yet they seem to have that option. Blood pressure monitors. What is the fundamental difference that lets them sell NIBP on Amazon and not cleared pulse oximeters? Is there anything preventing them from doing that?
No, so I don't regulate the blood pressure cuffs. I'm a little bit, I have to speak with some caveats here. But generally speaking, if something's over the counter, you could sell it at a pharmacy or, online retailer. So if it's over the counter, you could go directly to the patient. Now, not all devices, as people well know, are higher risk and it could be bought over the counter. Temperature monitors, blood pressure cuffs. There are a lot of other devices. Even the glucometers can also be purchased without a prescription. So depending on how the device has been cleared, they could be over the counter. It does not necessarily mean there's a difference in how accurate the technology would be, but it's established that for this particular intended use, that you don't need a learned intermediary.
But they could,
And I
they
think
could
along
still
those
be class
lines,
two. Sorry for interrupting, Julian. They could
Yes.
still be class 2 devices, even though they're over the counter, which would require them to demonstrate their performance in a 510K.
and regarding
quite-
not blood pressure monitoring, I mean, I would say it's even more complex to interpret blood pressure than it is pulse oximetry. It varies with as we know, right? Baseline requires sitting for 5 minutes without speaking. Blood pressure varies. Patients have a tremendous number of questions. Look at public forums where they don't know how to interpret their blood pressure data. So I think there's a model there that we should be using. It's unclear to me why we haven't been able to do that, but I believe this is one of the reasons that it's been difficult to put devices with appropriate performance into the hands of patients and empower them to purchase them and use them. So
Can I just put
Yeah,
my
David,
hand
over
up for a
to you.
while? I've been on the NIBP committees since they were Europe owned in 1958. We have exactly the same discussions in the NIBP committees about how to control the over-the-counter stuff that's sold for sports. And I don't think Gillian's quite right in saying that you can't buy it. I've got the Amazon page. Okay, it's a UK Amazon page, but I've got it in front of me and I can buy NIBP devices.
no, I was saying you couldn't buy pulse oximeters. That's exactly my point, that you can buy NIBP. They are available to the end user, even though they're somewhat complicated to use, and those devices meet standards and they're cleared. But when it comes to pulse oximetry, we have prevented manufacturers from selling those to the end user, devices that are cleared, that are FDA cleared. That's
Right,
that's exactly the point. Yeah.
And I can accept that you've had the controls to be able to do that, but it's not obvious. I got my pulse oximeters from Amazon. Yep.
Right. Well, it's possible, but it supposedly isn't permitted.
As did we all.
Yeah. Right.
And
anyway,
the other
thank
problem,
you for
while I've got a voice, the other situation that I have used pulse oximeters is when I go skiing, because that's voluntary desaturation, and I'm sure that if you, instead of offering to partially strangle your patients by giving them a low quality of air, offer them a ski holiday, and you'll be able to get most of the results you're looking for.
Thanks, David. We'll go over to Phil.
Just to be clear, manufacturers have stated explicitly that they would like to sell, they wish they could sell their devices the same way that NIBPs are sold, but they have not been able to in the US because they're not permitted to sell medical devices that are cleared without the order of a physician. So that's what they've told me. I've heard it many times. We've looked into this for our organization. It may be incorrect, but maybe that's part of what has to be addressed in this with this topic in the workshop. Thanks.
Thanks, Julian. Phil.
I just wanted to add a voice in support of what Julian's saying. I think an inaccurate pulse oximeter, one that fails to diagnose clinically significant hypoxemia, is at least a bigger risk as an inaccurate blood pressure cuff.
Sounds good. Thanks, Phil. A couple other, I'm just going to go back to this slide. We'll talk about this for a few more minutes, and then we'll move on to some of our other discussions. So this issue of expected performance to end users, if you buy a device, is there a better way that we could be communicating what to expect from that device? Is there a better way to communicate who that device has been calibrated in, what patient population, et cetera? There's package inserts and things like that, but some of the ideas that had come up have included, for example, putting a QR code, requiring that to be on all devices so that the end user can quickly access manufacturer data directly from the manufacturer about that, and that data would be current. if new data came out on testing that device, the end user would have access to that, even if they threw the box away 20 years ago, or 5 years ago, or 3 years ago. Is that something that would be useful, especially for Pulse Ox, where it's very dynamic, where there's a lot of variability between devices, and it also may be a way to communicate to the end user how to properly use the devices, et cetera, when that sort of information comes down the pipeline. But yeah, please.
Well, one part of the practicality. The reality is that a lot of scenario, we have a monitor from one company, an algorithm from another company, a cable from yet another company, and a sensor from yet another company. So which URL, which one do you take a picture of? Which instructions do you follow?
And you're mostly talking about multimodal devices.
Yes.
Yeah. So I mean,
We're talking about
this may be more pragmatic for handheld, benchtop, fingertip,
even
but
handheld
even though there could be
Well,
mixed match.
at least in the US, even the multimodal devices, the oximetry piece is all made by the same manufacturer. Well, that's, I guess, not completely true. You can get third-party cables which tend to screw everything up.
correct. That's where my brain was going.
Yeah, that's a fair point.
So,
yeah.
And also in terms of maintaining that, sensors get updated, cables get updated, monitors get updated, but cables may exist from the older groups. So try and keep a body of evidence that will be behind that URL that says, okay, we got this software with this monitor, with this cable, and with this sensor. It's impossible to maintain that for the user to be able to go, okay, I took a picture of the cable and the cable had the code I scanned in. How do I know that that's really attached to what sensor or to what monitor and what setting to know what my accuracy is or what the study is behind it? It'd be really hard to maintain that and make it usable for bedside nurse.
Fair point. Grant?
I think of the safe surgery checklist where, it was such a simple, useful tool. And there's a clear need to, this came up in discussion yesterday, it's as well today, about better training of the personnel who are using it. And that, a lot of that sits with the responsibility of the institutions that procure the equipment. But just sort of the foundations of better training, I think, is the starting point.
Thanks. Good point. Other comments on that in particular?
I've got just one brief, and it's maybe kind of simplistic, but I like the idea of the QR code. But I think the first order of importance on the QR code would be, it would say medical grade or not medical grade. Medical grade would be by whatever the FDA clearance standards are at that time, and hopefully they'll improve. That no pulse oximeter should be used for medical decision making unless it's a medical grade pulse oximeter. That should be the fundamental, I think, data that they would be able to acquire.
It's a good point. I mean, fundamentally, that is the biggest question: is how much can I trust this device? What is it intended to use? How can we communicate that better? People are clearly having a tough time figuring that out. Other thoughts on that?
How?
Yeah,
A lot. The global health side, which we both work in, we know a lot of that stuff goes into these low resource countries. So I think, yeah.
I mean, it depends on the market you're talking about, right? In the US, if it doesn't say cleared, or have a 510K number on it, you can pretty much guarantee it's not.
No, I'm referring specifically to, outside the United States in low resource countries.
Right, that's going to be a challenge.
Yeah, price sensitivity is the highest driver of what's procured.
Yeah,
Mike, go ahead.
having a little experience in that because of the lab that Phil and I set up in China. There's a big cost barrier to getting them tested, but it's not like the manufacturers won't. And one of the questions I'd just like to ask everybody is, what about having what I would call a mid-grade pulse oximeter, one that had a lower accuracy spec, wasn't considered a hospital grade one, but was tested, and maybe tested only in oximeter reference tests rather than blood, etc.
For low resource countries?
For any,
I think that this is forgive me if I'm a little bit passionate, but I think that we have a double standard and that we have a lowering the bar for low resource countries rather than raising the bar to say that low resource countries should have access to the same types of accuracy and equipment that we use or take for granted in the high resource countries. So I'm strongly against.
I'd like to add on to that just from a little perspective of that just because they don't have funds doesn't mean that they don't need the same level of care. So we need to have equity for care. We have to provide how we can do that financially. Maybe that's a topic outside this meeting, but for people, to hypothesize that because the individual is not in the hospital, that the decision that's made on the basis of the pulse oximeter is not as important as somebody in the hospital. Sometimes that might be not accurate because we can have really sick and complicated patients outside or even on general care wards that are in.
I completely agree.
We don't.
Hold on one sec, Mike.
I think I've made that point.
Over to Bob, then we'll go back to you, Mike.
I hear that there are resource challenges, and as a result, in order to have the product, minimally have the product, you need to have something that's readily available and low cost. I would bet that the bulk of pulse oximeter, inexpensive pulse oximeters, were being bought in this country during COVID because of the preponderance of what was on Amazon and eBay.
I don't know if that's true.
It would be good to get a pulse on that because they were selling like hotcakes. They were out of stock. And when Amazon's out of stock, you know it's a problem. Okay, so and so to Sandy's point, when he said, well, make sure they're FDA cleared, the vast majority of what you'd see on Amazon had the FDA logo on it. Now I know it's meaningless. Okay, don't get me wrong. The issue that I like to see is what you're referring to, Mike, and that is the manufacturer needs to have by QR code, by some other labeling, brightly on the device, not the box and insert, what are the accuracy specifications? And then it's up to the dear user, are they happy with this level of imprecision? They have to have the values of real testing. That's my suggestion.
Thanks. I'll go to Mike, then I'll make a comment. Phil. Yep.
So my argument about the lower cost one is that we have to incentivize that. There's no way that a government regulation can stop people in China from shipping something in and labeling it however they want. So we have to incentivize them to come into our lab in Shenzhen or some other place and actually test the thing. And the incentive would be if people actually started looking for these I'm thinking the LMIC, started looking for these, the people who are doing the big tenders started looking for this mid-grade because it would have a price that they could afford because they can't afford $400 or $500 per pulse oximeter, but it has some accuracy requirements.
I'm gonna make a quick point. I think now is not the time, this is my personal opinion, I think now is not the time to walk back and loosen standards. I think we have performance problems. We have inappropriate donations being made. If anything, we need to tighten down and double down our efforts to make affordable, safe devices, not create another category that muddies the water. Additionally, it would make it harder for consumers to know we're throwing in a third category. So I kind of echo what Grant had said earlier. The other piece of that is when we talk about lifetime costs, this is something that we've talked about. You introduce a middle of the road oximeter, a fingertip that let's say meets this intermediate standard. There's a pretty good chance that manufacturer has not put in the time to make it a durable device either, which means that the end user who receives that in a place that maybe didn't have the resources to buy the $500 device is going to buy that device 25 times over the course of 10 years because it only lasts about 3 months. It would have been much better spending $400 and buy the device that lasts for 5 to 10 years. And the systems, that's not a deal breaker in many of these systems. It's just that it's so hard to navigate and understand the differences between these devices that people make the assumption, whether it's decision makers and users, aid agencies, that the devices are the same. And so why not go for the cheaper one? And there are big differences. So I think that is going to create a big problem if we had a kind of a second tier. Phil and then Grant.
Yeah, I completely agree with what you're saying, Mike. So it is totally possible to make a pretty good pulse oximeter for $20. It sells for $20. The problem is not a technological barrier. It's an information barrier. It's knowing which $20 oximeters actually work pretty well and can tell if you're hypoxic or not.
Yeah.
That's what we've really got to change. And that information, like the QR code, that's a step in the right direction to my mind.
Grant, over to you.
Just a real world example. I was on a phone call a few weeks ago, and Rwanda received a donation of 8,000 pulse oximeters, and out of the box, 1/3 of them did not work. That's, straight up. And then these devices are deployed across a national setting. So you're paying for an enormous amount of logistics costs as well. It's not as easy as just calling up the manufacturer.
Yeah, I think we need to separate performance from reliability issues. I mean, they're vastly different and can be handled differently.
Thanks, Sandy. We'll come back to that. I see Dr. Mulroy, you have your hand raised. Over to you.
Must be a delay over to the UK.
Well, the delay may be that it's nearly midnight.
I think we lost you. Jake.
Yeah, just to bring it back to the topic of communication, like QR codes are great, but my mind went right to cybersecurity as being a potential risk, right? Like, what if somebody puts in a malicious attack on a QR code? It would be hard to control. Somebody snaps a photo of it in the hospital. Next thing you know, you have a virus or some other malicious attack. And so it would be interesting to know if there are other thoughts about how to communicate this information to the end user.
Julian, can you answer that? Since you're heading that effort for Amy?
John? We'll come back. Let's put a pin in that issue in particular. We'll go to the last one here, and then we'll move on to some other topics. We can come back if we have time. Communicating, better ways to communicate to the clinician at the bedside the confidence of the measurement. How can devices do a better job of that?
No, Bob, I'm here. I just could not get this thing off mute on the iPhone. So.
All right. Did you hear the question?
It was a little vague. Sorry, from what I heard as I was struggling with my phone. Could you just repeat it?
Yeah, go ahead.
I think so. Probably twofold, but the QR codes could present a cybersecurity risk. Is that something that's been looked into? And then, if they are, what would be other forms of communicating to the end user?
So those are good questions. So the idea of using a QR code to provide information at the point of care is something that we have promoted very strongly in a number of circles, including the Anesthesia Patient Safety Foundation and also at the ASA, and presented that and popularized it. And the APSF strongly supported it. And a few manufacturers have started including digital QR codes, for example, right on an anesthesia machine that you could scan and get information. As we all know, a QR code could be read and interpreted directly because the content can be put into the image itself, into the QR code, or the QR code could serve as a pointer to redirect you to a website or even a different website. The systems are pretty well developed and pretty well worked out, so I don't think that they present an undue cybersecurity risk inherently. But if you're going to be sent to a website and then sent to another website and so on, naturally that could be an issue. But the QR code itself could just carry pertinent information that you might need at the point of care. So if you're looking at a spec, if you're looking at other information, that could be encoded and read right from your smartphone, and there is no likely cybersecurity risk. Now, there's always games people have played with encoding something in a QR code or doing something like that sends a command to the phone. And that's pretty kind of out there in terms of security. It's possible, it's been proven, but I don't think that should be the focus. I think it's more about which website are you being sent to, and is that appropriate and safe? So it's a superb solution. No more risky typically than going to any other website. So all the usual controls would apply. So good question.
Thanks, Julian. I see Dr. Mulroy has joined back on. If you still have a comment, please let us know.
Hi, can you hear me now?
Yes, we can hear you.
Brilliant. It was telling me my microphone wasn't working, so I came out and in again. So hopefully that's fixed it. I just thought it was worth raising that in the UK, there are 4 classes of risk device for devices from 1, 2A, 2B, and 3. And within that, so devices for continuous surveillance of vital physiological processes, anesthesia, intensive care, or emergency care are considered class 2B, whilst those intended to be used to obtain readings of vital or physiological signals in routine checkups and self-monitoring are 2A, but these would both require the oversight of a conformity assessment body as part of the conformity to the regulations. It wouldn't be the MHRA looking at that, would be the approved bodies, conformity assessment bodies. I didn't know if that was useful or interesting.
So does both 2A and 2B devices have to conform to the ISO standard?
No, they don't have to. They can use that as a way of demonstrating safety effects, but they don't have to. It's not mandatory to conform to the standard.
So neither 2A nor 2B have to be, quote, medical device. How else can one demonstrate conformance?
They meet the requirements of the regulations, and I mean they can use the standard, and I know it's the most. What else are they going to use? But it's not it's just in terms of the process, it's not mandatory.
Yeah, can you talk EU as well, or are you just UK?
Sorry?
Is it the same in the EU? I thought in the EU they had to demonstrate conformance to the harmonized standard, the ISO standard.
If it's harmonized, then they can use it to demonstrate that they meet particular requirements of the regulations.
I think it used to be. I don't know if it still is.
It's still not mandatory, but if it's harmonized, there's a kind of more closer connection with showing.
Well, so I'm sorry for monopolizing this, but so demonstrating that you conform to the standard is implicit evidence that you conform to the regulation, but it's not mandatory, is what you're saying.
Yeah, but if it's a harmonized or designated standard, yeah.
But that's the quickest way home.
Yeah, if it is, yeah. Harmonized, yeah.
Okay, thanks very much for sharing that. Appreciate it. So that the last bullet point here, closing that out, any thoughts on how devices could better communicate to the clinician at the bedside?
Maybe we could start with a description of what's currently required about that standard.
Sandy, can you read what's in the current standard by chance on that issue?
And Julian, if you want to emphasize the interoperability side of it for communication even to an EMR.
Let me share my screen for a moment. And
you're gonna, Sandy, you're gonna show the annex?
You want to do that? We can do that as well. We can do lots of things. Do you want to? Am I sharing?
Yeah, I think you are.
Do you want to do the annex first, or do you want to do labeling first? I lost where our thread was.
It's about what the standard requires for advising the user about signal quality.
Mm-hmm. And whether or not that's captured in the record.
Now, I heard 3 things now. All right, so I think what we're talking about is what information the standard, and it's not normative, it's informative, suggests be broadcast out. It's a SIP-SOP, signal input, signal output port. And as soon as I can.
You had it, Sandy, you showed these, if you search for the Society for Technology and Anesthesia.
This is what we're interested in, right? The data interface requirements?
You had it there before. You had the STA there when you were searching.
So I'm confused as to what you're looking for. If we're looking for the information to be communicated electronically, that's this information. You're looking for something else?
Well, signal quality. What does the user know about the quality of the signal? What's required to be disclosed to the user?
So
not the or other data acquisition system.
So we have a clause called signal inadequacy, which is inadequate. Right, and you can kind of read, right? It says an indicator of signal adequacy shall be provided. And then it points you to the accompanying documents to explain what the heck you mean by signal inadequacy and how you tested it.
So I think just to, and Sandy, if you search for STA, so I think we're talking about a few different things, and that's good.
Yes, we are. That's
So
why
the
I'm
notion,
confused.
right. So the notion of what information should be presented to the user or operator, that's what Sandy was just, talking about, that signal inadequacy is the way that it's handled in the standard. The other topic is what information should the device transmit or emit from its interface, its electronic interface, in order to, do all the things we know must be done, whether it's data acquisition for research, whether it's to transmit data to some type of electronic record keeping system, and so on. So the standard is referencing something that emerged from the Society for Technology and Anesthesia, which is essentially high-level guidance. It doesn't have the specifics. It points out that anything that's displayed on the screen to the operator, which is used to operate the instrument, interpret data, that same information should be transmitted through the electronic data interface. So I think a good example would be things like perfusion index or signal quality index, things of that sort, that you'd want to have available so that you could then analyze the data later. So Mike, you said you don't agree with the statement. Is it this statement or the previous statement about signal inadequacy you were commenting on?
Am I doing? I was just writing in the chat. I don't agree with the statement from David Scott. Sorry, a different thread.
Oh, sorry. Okay, no problem. So this is, as you can see, this is pretty high level, and everyone could see it. It talks about things that should be available through the electronic data interface or information about the device, including software versions, the mode of operation or state of the device. So is it active? Is it processing? Did you change the signal average time? The clock time, pretty important because there's lots of errors due to things not being synchronized correctly in data. So that's this high level statement. Now there's a more detailed analysis that is also included in the standard in Annex, was it B or can't remember, Sandy, which enumerates some of the desired outputs from the device. And this work is being actively expanded for a whole range of devices. So, this is there's an opportunity here for this group of experts to help enhance the list of desired information that you would like a pulse oximeter to emit from its interface. And I would propose that work be, we've done some of that work at MGH as part of our interoperability efforts. And it's certainly a good place to park that information so that it has traction is in the standard. So I don't want to take too much time. There's a lot going on, but hopefully that addressed the points that were raised.
I don't think that quite got to my point. I mean, I understand how the data transfer is really important. That's kind of a separate topic, though, from what I was bringing up. What I was asking about is what in the standard, if anything, and I think there's nothing, it's just really vague, about the quality of the signal that the operator sees. How does the operator know that the measurement is reliable? What kind of reliability indication accompanies the pulse oximeter estimation of saturation?
Yep. So Sandy can bring up the section on signal inadequacy. And I think the reason it was written that way is it reflected the fact that people have different approaches and manufacturers have different approaches to determining if they can trust the signal. So Sandy, I think you and others can speak to that. I mean, if there's a better way, clearly.
Right. Over the course of the last 3 decades, everybody's had different ways of describing signal inadequacy. And so that's why there is no consensus. By all means, if we can come to consensus and develop a test method for what this should mean and how it should be presented, I think that is a major step forward and we should do it.
And if I could, I think yesterday I added to the comments section something related to this, which is that manufacturers historically have said, have had to deal with competing requests from clinicians, which is hey, why are you showing me data if I can't trust it? You should be blanking the display. That's that one end, one extreme. And the other end is show me any data that you could possibly acquire and display, because that'll help me in some way. It may help you move a probe to a different position, to optimize the signal quality, for example. And so they've had to contend with this. And I think they're all valid points. So the better the signal quality indicator or signal inadequacy indicator is, that the more likely that we won't misinterpret the data that's being displayed.
Thank you. Look at, or no, look at this out here. The disclosure requirements. Really basic.
Mike, did you want to make a comment?
No, I was saying it's basically disclosure requirements. What's in
Well,
there?
yes,
Yeah, that's
much of our standard is a disclosure requirement because we couldn't agree on what to put in there.
From a clinician perspective, I feel like a lot of clinicians, I know certainly this is my own bias, but if I have a device and it doesn't display a waveform, I would prefer not to use it, because it gives me it's something that is easy to understand. Even if you're misunderstanding what it actually is telling you, people understand that it looks like a heart rate. It looks regular. It matches with some other parameter that I can validate in another way in that patient, which is the ECG heart rate or me feeling their pulse. It should be regular or irregular. I know what a normal waveform should look like roughly. It's a trainable thing. An example that I was talking about from the lab earlier, which is even a bit subtle, we were playing around with some of our handheld devices. Some of the probes can work with devices that they shouldn't work with. The only way that I was able to prove that repeatedly is that the waveform had a bunch of noise in it. And I was trying to figure out why. And it was because it was a cable that didn't work with it. But that was the only reason I knew that it didn't work. The waveform is extremely useful. It's an easy tool to educate clinicians on. It conveys a lot of this information about signal quality, and is kind of, the data is universally there. I'm speaking as a non-engineer, so forgive me in the room, but that seems like, I don't know. I wonder what are the downsides of requiring that a PPG waveform is part of the display. We'll go to Paul and then over here.
Well, the problem is that most waveforms today are filtered, so they look smooth, even though they're noisy. If we had some sort of quantification of around how much you can't smooth it, then
So
that would help.
unprocessed waveform, which I think Melcor is unprocessed waveform.
Non-normalized.
Non-normalized.
Non-normalized, yeah.
Non-normalized. So standardizing that in a way could be really useful. If it's auto scaled on the screen, I'm not really sure what to do with it. Or maybe I didn't even know it was auto scaled.
Yeah, so many of them are.
Right. So
And what do you do with the handheld devices that you don't have the real estate?
they can have the real estate. That's proven by many of them. But another side issue. Yeah, go ahead.
We want to just bring it back full circle and say, for you in your clinical judgment, you use the waveform to help qualify the signal quality and strength, but you're a trained clinician. We have products that are in the home that don't have a waveform, and we're now expecting them to make a judgment call on their care with even less information than you asked for as a clinician. So why are we having these two standards when you're making decisions off of effectively less data? I think it's even more argument that for the home care, if this is an issue and this is people's lives on the line, we've got to hold everyone to a higher standard and not lower the bar. So our wellness devices, accepting that they're going to be of lower quality, and that's a risk. That's going to cost some people their lives.
Thanks for the point, Carolyn. Oh, Carolyn.
I was just gonna say, I think in like modern care delivery, like I have people take pictures of their monitors or like hold them up to the screen to show me what's going on, and I can help interpret, even if it's a home device. That might be better than nothing. I'm not suggesting there should be different standards, but I do think people get support in interpreting their home data. It's valuable.
I mean, I can tell you we had a clinical panel meeting, just like we had, a few months ago in the mid 2000s to address home use oximeters. And the primary issue was, how do you write adequate labeling for the lay user to use the product safely? And the manufacturers, when they went to do their human factor studies, they said, look, we can take the batteries in and out. And I think you kind of missed the point of the cognitive ergonomic piece.
Yeah, but I mean, I almost want to, I kind of seeing, maybe I'm not seeing what you're saying, Sandy, but I'm almost seeing that as a separate issue. We're talking, why can't we make a device that works as well in the home setting as it works in any other setting?
Well, I think the performances, but we also have to include adequate labeling so that the lay user can use it safely. That's part of making a home device.
What if you could make a non-invasive pulmonary artery pressure cap? I mean, like, there are limitations to what people can do in the home care. We have got to acknowledge that. I mean, it's, but sorry to speak hyperbolically. Paul, did you want to make a comment?
Yeah, one of the things that I think is a showstopper that we've talked about a little bit earlier is the manufacturers, many, maybe most, think you cannot sell a clear device over the counter, a pulse oximeter over the counter. And if you could, if a lot of our good, well-performing devices that are medical devices were sold over the counter, that would help a lot. And they could, explain this has FDA clearance. That's a big deal. But right now, there's a hindrance, whether it's just perception and they should be putting it on over the counter or not. That's what's happening right now.
Sounds good. I'm
That's a
gonna go to
good
Bob.
James question. Why didn't you ask that when James was here?
Why don't you get James on? Because that is a real basic issue.
Well,
If you
James is busy doing other things. That's why.
Jonas will come to you after Bob. Okay, one sec, Jonas. Bob, go.
Yeah, so 2 points. I think we're really circling around the issue of education and interpreting what values are coming from the device. And a value is a trend display. It's also this active real-time PPG or some surrogate of it. So having that sense of signal quality indication is their value to the actual displayed measure becomes exceedingly important. And people are not
mandatory. See that?
Sorry, Jonas.
David?
Just gonna put you on mute.
Jonas?
Keep going, Bob.
Okay, I think is exceedingly important. Now, we have experts around the table, and I'll lean on one to start with, and that's Mike Jaffe. Mike has created an encyclopedia of end-tidal CO2 waveforms. How do you interpret it? Now, a PPG waveform interpretation, provided it's real, could be tremendously valuable. And I mean, we're only talking about 2 or 3 traces. So I think that could be very valuable. And we have the skill set here.
Try to do it. But for the purpose, just to refocus us, because we have limited time,
Yeah.
the purpose of the discussion here is not, should we educate people more? Is it feasible to educate people more? We all agree on that. I don't think that that is relevant to the standard, though. The standard, our question right now is, do we require something different in the standard to help facilitate this?
Right now, it's
so
open.
Not the question of, can we educate on waveforms? Can we make the material? Do we require waveform in every device?
No.
But I think the standard requires a signal inadequacy
That's right.
indicator. You can choose how to do it.
That's right.
But you
Does
have to
anybody
have
feel
one.
that signal inadequacy section should be changed in light of conversations the last 2 days?
I do. It's extremely vague. It gives manufacturers way too much leeway to provide information that's maybe
How can
not
we change?
useful.
I'm going to pause that for 1 sec. Jonas, you've had your hand up. Let me go to Jonas, then we'll continue on that conversation. Jonas, floor is yours.
Thank you. I just want to throw out the comment as a manufacturer that anytime we put in a requirement, we also are potentially limiting the capability of the device to provide for other needs. So I know this might be a silly example, but I think it gets the point across. If we said that all capnometers had to have a waveform, well, somebody made a capnometer that we use in emergency medicine, which consists of a little color disc that changes color as the patient breathes in and out, so that we have some sense that we're actually in the trachea and not in the esophagus. So had that have to have the requirement that it has a waveform, that never could have been brought into existence. So I bring that up simply to say that I would like us to be very cognizant of the fact that when we add a requirement, whether it be that it has to have a waveform or it has to have a signal quality indicator and a signal poorness indicator, we're putting constraints on the design and on the products themselves that might hurt us in terms of going forward.
Jonas, thanks. That's a great point. I'm noting we have 45 minutes left in the discussion. So I'm going to put a pin in this part. If everybody could think about, if you think there are things that should change in that, perhaps not, we can leave it. But when we come back at the end, we'll go around quickly and everybody will say their 3 kind of points that they want to make. So if that is one of them, just start brainstorming what you want to suggest to change discreetly for signal inadequacy in the standard. I'd like to propose going back to the issue of skin pigment quantification and completing our discussion of that, because I think that's really one of the core issues of the last 2 days, and try and develop some consensus on how to move forward on that particular topic, if that's okay. Anybody object to that? Okay. To start it off, Sandy, can you just read for everybody? I think everybody's very well familiar with it, but what the current standard has in for diversity requirement? Not much.
Pretty easy, basically nothing, but let me.
Gender and skin pigmentation in 2 buckets. And gender's in 2 buckets too. Sorry.
Okay, that's okay. While Sandy's pulling that up, and just ask for hands, people to make a proposal for what that should change to, and we can go around and hear those. Bob, did you want to make some comments?
No.
Okay.
Can you see my screen?
Yes, we can. Yeah.
See, it's pretty easy. It's only color. You have to be British.
Color.
I had pasted that into the chat a long time ago. So this clearly is right for fixing.
Thoughts, comments on this particular section?
And realize this is currently still informative. So this is not a requirement. The place where it gets required is in the US if you want FDA clearance, because it's part of the guidance.
I'm sorry, Bob, that's not. This is normative, this section. The desaturation protocol is informative. But this section is normative. And as Bob points out, this has really been modified by the FDA guidance document, which has slightly more information. And I would like to point out that, if we can't come to consensus as to what we think this should look like, everybody's going to do it a little bit differently, and we're going to have mass confusion. I think I might have already said that, but so anyway.
Floor is open. Thoughts, comments on modification of this section? Raise your hands. Bob, you want to lead off?
Well, Leo, I think is more of an expert in this place because, yeah.
Well, I'm happy to share. I don't know what expertise I really have other than being able to raise questions and saying I don't know the answer. I think some of the things that I think collectively there's been a lot of agreement around over the last 2 days is, number one, using some sort of standardized scale that explicitly includes a broad array of human pigmentation. So, not just leaving it open to say color, but making explicit that we need to use some sort of scale that includes pigmentation from light pigmentation to dark pigmentation and representative of human pigmentation. I think that, my personal bias, I'm sure all of you can guess, is that of all the scales that are out there right now, we should use one that exists and that is well characterized. And the fact that Google and Facebook have adopted the Monk scale, and there's been studies showing its utility and its applicability, I think it's totally reasonable to start there. I don't think we have to limit it to the Monk scale. I think, other scales can be made in the future. Maybe the Monk scale gets updated in certain ways. We should be open to that possibility. But I think some sort of objective color scale that is representative and includes pigmented skin in a more complete way than historical scales have is an important next step that should come out of this. I also think that the percentage of 15% needs to go up. It is just in the current age, even in the United States, that is not a representative number, and across the world, it is not a representative number. I think that it's important also to keep self-identified race.
Wait. Can you go back to number of subjects per bin? Are you suggesting that it should be an equal number of subjects per bin, or it should be reflective of current demographics?
I think at minimum reflective of current demographics. I don't know. The problem of subjects per bin is I don't know exactly how many subjects there are in each bin, and so I'm not sure about that. And I'm open to other people's ideas, and Ellis may have a better answer that's more intelligent than what I've just said. But I think I just wanted to make sure also that in our efforts to include a color scale that's more subjective and complete, In doing so, we are really focusing on skin color, right? And as we've heard from anthropology's talks, which I think were really excellent in expanding our view of this, and Alice's talk yesterday, color itself and race, these are not necessarily connected. One is a social construct, one is something we see. And it's important that we maintain in our studies of this question that self-identified race and ethnicity is an important component. And we should not assume that by measuring skin color, we automatically can guess what someone's self-identified race is. And I think that that's a really important thing, because while, I think the work that Chetan has presented is incredibly powerful from a bio-optics perspective. As a physicist, I love it. There is still the question, the possibility still exists that all of these things are factors, that racial health disparities is a factor, that melanin scattered absorption is a factor, that all of these things contribute, and we should not close our minds to that. So I think keeping those measurements alive is important. I don't know, I said a lot right now, so I should probably stop talking and see what other people have to say, but those are my initial thoughts on the matter. And later, I'm assuming we're going to get to like, what do we actually measure? But this is just the question of what do we say for inclusion? And again, I can talk about the spectrophotometer and all those things as well.
Yeah, let's come back to that. Other comments on anybody? Chetan, I see your hand.
Yeah, I was going to add to this by asking about the discrepancy or drawing a distinction between asking the manufacturers to describe how they've constructed their model and then tested their model, because I think the thing that's most evident to me is that you can test any sort of model a number of different ways, but the rigor with which you need to test the model, I think, is informed by how much you know about the model's construction. So
I'm a little confused. Sorry for interrupting. What model are you talking about?
Well, the tests that we're talking about here are testing a certain oximeter calibration model against some data that the companies are collecting, right? So it's informative to understand how they've constructed the model.
Chetan, are you talking about the calibration curve between R and SpO2?
Yeah,
when you say
okay. Yes, the
like
calibration, I should say.
Curve that's being used.
Yeah, so that's just it's another element that's not in the that's in the standard, but I just thought I'd bring that up because I think one informs the other.
And I'll report that, to date, manufacturers have very tightly guarded their calibration curves because they think that's their secret sauce. And I'm not sure what's going to happen if you try to mandate that be, demonstrated or, revealed. So
described. I think then the alternative is that, maybe you ask for testing to be done on separate cohorts at the extremes of whatever color pigmentation standard is used, because that may be the most rigorous way to test it, let's say.
And I'll reiterate that, and I'm not a manufacturer, right? I'm just a regulator, but manufacturers, I believe, do a lot of development work. And at the very last stage, they're doing what I call qualification, which is assessment on a relatively small number of subjects to verify that their device performs according to specification. Right? So they're not trying to validate that their product meets a particular clinical intended use. There's a difference.
Right, so the term verification, Sandy, do you wish to make that distinction?
Well, I have been trying to, yes. So verification is demonstration that you meet a particular specification. And in the US, we use roughly 3% as the specification, right? Your ARMS has to be less than 3% to get on the market. That says nothing about the clinical intended use of their product. there is an implication that your product is clinically useful if your ARMS is small enough. But I am not trying to show that my device is able to detect COVID or ARDS, and I'm an engineer, so I'm speaking things I don't know what I'm talking about, any other clinical condition. Or in fact that a clinical uncertainty of 3% is acceptable, right? It's a very tight engineering specification that I'm trying to verify that I meet.
Okay, so there is one point I'd like to make coming back to your suggestion, and that is if we're trying to represent the patient population at large, we're clearly not doing it with the 15% bucket of darkly pigmented individuals.
Right.
And I can go city to city almost and say that this is a rather dynamic situation. But we ought to come up with a median and say 15 is not doing it for dark, and we're underrepresented for brown, and the Caucasian population is overrepresented. So I think we can at least come to a decision about how those.
Well, short of just, I don't remember exactly what the demographic numbers are, but there are demographics that are known for these values, and I think it should mirror the representative demographics of the country.
Country or the world?
Well, I would say world, but since it's FDA, I don't know what the rules are here.
Everybody in favor of the world versus the US. Everybody in favor of the world?
I'm certainly in favor of the world, as long as we're allowed to say that.
All right, let the record show. Sorry, Carolyn, comment to Carolyn. And then over to Bill.
Frame this question. So I think when you're talking about representation and leadership positions and who's making decisions, the demographics should reflect the population you're serving. I think in this case, we have to be able to detect a difference that's clinically relevant for the people who are most at risk of being underserved by this device. And so maybe it's not that it reflects the demographics of the population. Maybe we have to do some power calculations to make sure that we have enough darker pigmented skinned people to tell us if the device is performing differently for them than for people who are of lighter skin pigmentation. So I don't know that just translating the census demographics of the world or the US to our study populations is actually the thing that's going to keep people safest or make us feel okay about our technology.
Let me clarify. It needs to cover the range of skin colors in the world, not the range of skin colors in the outer sunset of San Francisco, where we draw on our study population. Sorry, to clarify, I think that was the point. I think we run the risk if we make it in a certain country for a device that is applicable globally, that we run the risk of.
But I think Carolyn
is that consistent with the point of
our calculation? So I think there needs to be some sort of text about a defined number in terms of how many testing devices.
The problem is we don't have enough data to make an informed power calculation.
I'm not sure that's correct, Mike, because if you look at the
I haven't been convinced by anything.
if you look at the work that Gene's been doing, Gene's been looking at the different groups, trying to determine whether or not the biases is different between the groups.
Sample of 10 to 3 people. That's still not enough.
Agree with that.
Hang on one second, Sandy. I really wanted to make sure a point that Carolyn had brought up. Ask about James. That one of Carolyn's point is well handled. I'm not sure if we're going to get to the top 3 thing, but I want to get one off the table, which mirrors where she's at. How are these devices being used? They're being used a lot in situations where people at risk are not getting the correct numbers. And that's in home care, it's in their person. And when I say home care, it's the emphysematous patient at home or the cardiac failure patient at home that is looking at this device and they're becoming non-compliant to their drugs because they're hypoxic. Now, that's crazy. And why? Because we're not testing on some of those individuals as well. So I think the testing that we may be doing, which is verification, final step, exceedingly important. But if we're not doing convenience sampling on some of those type of situations, and the device is not working there. And that's the intended use. That's the place where it really matters in life changing or life support, where they're getting oxygen, etc.
Okay, so Mike, we got a lot of people with hands up.
Thanks.
Raise your hand if you want to speak, please. Bob, anything else to say on that?
No.
Okay, we're gonna go to David Scott, then Ellis, then Phil. And then Mike. David Scott, go for it.
Right, thank you. What concerns me is that Jeff Matthews has come up with an answer to the problem. It seems to me to be fairly easy to verify whether or not the LEDs are working on the right wavelength. And until you've got an answer to that question, everything else is irrelevant. So please assure me that somebody else is going to try and verify that work.
Thanks for the comment, David. Ellis, we're gonna go to you. We're trying to stay focused on this. Yeah, and then we'll go.
Okay, so really quickly. So first of all, I want to completely echo what Leo said. I do think that, first of all, the Fitzpatrick and the von Luschen scales, I don't really see a place for their continued use. Of course, I'm Ellis Monk. I'm going to tell you I recommend using the Monk scale. But one thing I want to say about the kind of worldwide distribution of skin tone, which is why the scale is the way it is if we look at these maps, the one on the left, figure 3, is from Jablonski at Penn State, who's a worldwide leading expert on skin color at Penn State. We can see that most of the skin tones in the world are not going to be very light. So if we look at what we're testing, and I want to echo what Carolyn said here, really important point. If our hypothesis is that melanin is something that we need to be considering here, and historically these devices have not been tested on a diverse range of patients and study participants, then it may make sense, and I think Medtronic even said that they did this, to actually oversample people who are darker skinned, not just to have equal bins, but rather to make sure that you're going to have people who are darker skinned as well as people who are lighter skinned, to kind of elevate what Chetan said as well. If we believe that melanin is something, you know, at issue here, it might not be adequate to just have equal bins, but rather to even oversample to make extra sure that we have darkly pigmented people in these studies. So elevating that 2 or 15% recommendation. And then when we bin a scale, if it's my scale, for instance, you could bin my scale into even 5 bins to avoid the 6 bin confusion with the Fitzpatrick scales of the past. Make sure that in that 5th bin, if you want to do 5 bins, that you have, if you're going to do equal representation, fine, but I would suggest maybe even bump that up a little higher, just to make sure we're including darker skinned people into these studies, to make sure that these devices work equally well across the entire skin tone continuum.
Thanks, Alice. We'll go to Phil, then Paul, then Mike.
So I have a specific regulatory way to think about this, I think, or suggestion. And that is that the manufacturers need to show that the bias in their devices does not vary between different classes of skin pigment. If it's just that, then they would be required to have a statistically powerful enough sample of the darkest and lightest and intermediate skin pigment. So it automatically kind of solves the demographic distribution question. If the requirement is to show that the device works equally well in all types of human skin pigment. It was just simply that. To achieve that would require that distribution of inclusion.
Thanks, Phil. Paul, then we'll get Mike.
Okay, so three things. Pigmentation, site measurement, methods, and then sample size for differentiation. First, let me just share this. All right, here we go. Okay, so self-identification of color is generally related to the person's facial countenance, and thus for the personal societal recommendations, I think we should collect the information from the forehead, which is the area that of the facial countenance, dermatological site for assessment of the baseline pigmentation. I believe Leo's made it clear that the inner upper arm is one of the places that should be measured, and I think we should measure that perhaps for the bucketing aspect of the baseline pigmentation of the individual, and then sensor site. We don't know what the influence of that is. Data that we've looked at a little bit earlier today that I shows that there's doesn't seem to be a significant difference between the light finger and the dark forehead of the same individuals. So I vote for those three sites as being the where we measure. I think we need an objective method of measuring, and I think Wim and Leo can guide us in that. I think we should also use the Monk scale when that is not possible, as we've talked about in some areas, and times when it's not possible. And then the third thing for sample size, I'll go up to slide 23. And so some of the information that we've collected, this is from 80,000 cluster bootstrap strap samples of sample size 30. It's kind of hard to see, but each of the clusters were 30 subjects with 10 darkly pigmented subjects and 20 lightly pigmented subjects. And as a sample size of 10, you'll see here, you can look all the way down to the bottom. It's kind of hard to see, but there is the RMS results for and the distribution.
How did you define dark and light in this?
Yeah, and that was the 5 color assessment. It was visual. This was done over a 3-year period, a few years back, from 2017 to 2020.
And dark is which of those 5?
And dark is the well, I'll show you that.
Okay.
Yeah. And so we're just talking about sample size right now. And a sample size, you can see the spread of the data, and the tails are much larger in a sample size of 10. Sample size of 30, it begins to get more reasonable with the tails somewhere between 1.5 RMS and less than right around 2.5. With a sample size of 81, then it gets tighter still, and then with a sample size of 150, we see that get even tighter. Now, quickly going to the next slide here. So this is the differentiation in colors: the light, medium, and medium, dark, and dark. And we collapsed the medium dark into the dark because there was so much more dark, which we would consider a Fitzpatrick of 6, than there were of the samples from the medium dark. We put those together, and the linear model was fit to this, and we found that statistically the differences in performance in this 30 subject bootstrapped groups were significant. And this is kind of, to put it into my kind of terms, basically. So this shows that what the light, medium, and darker skin colors would have read at 70%. And the differentiation here is slight, but it was statistically significant. So this is beginning to give us the idea that a sample size of 30 is meaningful. And if we had 30% of these, though, were darkly pigmented subjects, and the total was a 30 sample size. Now, this was analysis done by Gene, and he found with if you're differentiating from sample size of 3 groups, if you differentiate in 3 groups, where we just differentiated in 4 to 5 groups, but 3 groups, your sample size needs to be around 32 at the bottom end, and for a much less well-performing device, a 46. So that's where we are with the empirical information on a fairly large data set in a comparison to co-oximetry. So that's my story, and I'm sticking to it.
Thanks, Paul. I'm sure we'll have lots of comments on that. Mike, did you want to make any comments?
Actually, I wanted to say the exact same thing that Paul said in a different way, which is that if we wanted to be as sure about how it was going to work on a dark subject as everybody else, then we would have to test 10 or probably more dark subjects.
Yeah. But I will take issue with one thing from Phil, and that is we can't say they're equivalent. We can say that they're within the performance specification.
It's a statistical
Yes.
concept.
Yes, I agree, and that's probably what you were wanting to say. Yeah.
And I would maybe just comment on that as well, as like, what would be the difference that you'd want to be able to see? Or when would something be meaningful to you to be able to tell a difference in terms of bias if you're looking at
Are you asking what's clinically significant?
Yes.
What was your set to predicting?
What's that?
What was your model set to predicting in your power calculation?
That was 80%. I think that's what
To detect what difference?
Yeah.
to detect difference in 3 groups. Yeah, well, that was 80% was what Gene used.
But what
For the power.
SPO2? But what SPO2?
What's the effect size?
2% different? Yeah, what's the effect size?
Yeah, and it was less than 1%.
That's pretty stringent.
That's pretty strict.
That's really tight.
Well, that's what was found in this empirical set of data.
Okay. And that's for all groups then?
Yeah.
It performed within 1%?
Yeah, let me just show you here.
All right, and while you're bringing that up, I bring that up as well because Bill, you had said, let's set the bar at 2%. And right now ISO says 4. It's been
We're
kind of
talking different bars, Bob.
Yeah, talking different bars.
Okay, and you know I don't drink. All right, so 4% is where I'm only bringing that up because 3% is
We're
what.
talking about bias, right? You're talking about 1% bias. The
Okay,
difference of
right.
1%
Yeah,
performance difference.
Yeah, so
Not ARMS.
if you look at the screen, this shows at 70%, the difference was 0.5% among the groups. What the device would have read there, and at 97%, it was like 0.7%.
Okay. So 1%.
Yeah.
Okay. All right. Thanks for answering.
So Mike, by the way, we have about 17 minutes or so before the hour. We were going to go and do our top 3.
Yep.
Is there still time for that, or should we?
Yeah, we're still going to do that. I'm anticipating that we are going to run over a little bit on time, so apologies, but we will still do that. But I think we got a couple more minutes on this discussion before we go to the round. Thanks for the reminder. Leo, were you going to check? Did you want to say something?
I just wanted to say, in terms of the objective measures, I don't know that we know for certain that the colorimeters that are kind of narrow band devices give us a complete set of information, or as complete information as you get from something like the conic minolta, which is a more like diffuse reflectance measure. So I think if possible, asking for us, since we don't know, to get the most complete data when we're measuring site-specific pigmentation measures, I think it makes sense to say we should measure it with the conic minolta. And I think that because we're potentially measuring small areas, picking an aperture size that is small enough to fit the finger makes sense, and to be standardized around that. And then, the software allows you to extract LAB values from that reflectance measurement after the fact. So you don't have to necessarily specify D65 or 10 degrees, but for people comparing values, I do think we need to specify a illuminant and observer as part of the standard. I will say though, I say this, I'm not 100% certain if you have to measure the inner arm with a Konica Minolta as part of inclusion studies, because I don't know exactly what you do with that information. I think that for the purposes of ensuring that we have people with darker pigment and lighter pigment, the fact is that the subjective scales just use facial appearance, right? No one's saying, hey, we got to do a Monk scale on the forehead and a Monk scale of the inner arm and a Monk scale on the finger. That's kind of a nonsensical statement, right? So when using the subjective scales, if we're using appearance, let's use appearance. And when using the objective measures, let's use those to measure the site-specific areas that are actually being measured for pulse oximetry. Output a diffuse reflectance spectra. I think it's natural to convert that into an ITA for ease of use, but knowing that there's limitations to what ITA really means. And ITA, as I showed, might just be a brightness measurement. It literally might just be L-star. So I don't want to like pigeonhole ourselves into a set of analysis before we know fully what it is. But I do think there's a differentiation between subjective scales used to ensure coverage of the full spectrum of populations. And by the way, Carolyn's point about making sure that we increase the numbers for the patients who really need it, I think is absolutely correct. But I don't think we need to measure someone with a Konica Minolta to do that. I think we can get there without that. It's the trying to get access to the optical properties, which diffuse reflectance is not perfect for, but it's the most complete answer I think we can do for now at the measurement site. So those two things are kind of different ideas, and I think both should be included.
Yeah, so the issue there, though, is if we measure at the measurement site, we're probably not going to get anybody or very, very few people in the darkest area, because the Palmer site, if we're looking at the finger, doesn't have that.
We shouldn't use the finger pigmentation as an index for whether you are in bin 10 or bin 1. Right. I'm not saying that.
You guys are talking about recruitment
Yes.
being separate from scientific
That's what
hypothesis testing about pigmentation interaction with the technology.
Exactly.
Right.
Those are the two things that are separate, and I don't think we have to overburden recruitment with you have to use this device that has potential questions about what site you pick and all this kind of stuff. Recruitment is, as Ellis has shown, these are scales that clearly differentiate people across a wide spectrum of pigmentation. And that's a recruitment question. And then when you talk about when you're doing these studies and you're validating it using a tool that gives you as much information as possible from an optics perspective, that is also not something that comes in a lab on a bench that you have to tweak and find students to fix for you when the device breaks, but a commercial device that really you can use. That I think the Konica Minolta or a similar spectrophotometer. There are other ones, the X-Rite, for example.
Best to rate it.
Right. But again, those are two different things. I think both are reasonable. If it turns out that a simple colorimeter could do it, or just as was discussed, just the red channel alone, maybe we just need to look at the red channel, the LED. That's all we need. That'd be great if it's that simple. But until we know that, I think getting as much data as possible in the standard makes a lot of sense.
Sounds good. In the interest of time, one last comment in the interest of time, we'll start going around and then we can.
Yeah, I'm a little bit confused. So if we want to ensure in the recruitment process that we have a spread of a population, for what do we then need other than understanding from this engineering point of view, the site measurement? So would we then basically do a statistics on if I am a finger sensor manufacturer, and I have clearly no differences in the pigmentation. So what does it tell then me, or what do I have as a manufacturer to report out as a standard? So I'm not clear what the standard requirement is to then claim to be compliant to the standard if we differentiate between recruitment and the analysis.
So what I'd like to say to that is I think we should measure in the clinical lab. I think we should measure all three sites, wherever the sensor is. I think we should measure the baseline pigmentation, and then for societal inclusion, the forehead, and then we can decide. But you're right. He brings up a point. Regulatory submission is going to say, okay, did you have this level of inclusion at the sensor site, if that's where we're requiring it, or are we looking at baseline? Generally speaking, that's the color of this individual.
And I think actually for recruitment, you say overburdening the recruitment. But once you have the colorimeter anyway to measure the fingertips, you might as well use it for recruitment as well and for inclusion and binning, because actually the color skills, if you want to standardize that's quite a burden too. I mean, we ask for an open window with daylight coming in, and you have to have an operator. You have to sort of standardize that. It might be a lot easier to use actually the objective measurements for recruitment and binning and inclusion.
In the clinical lab for regulatory submission, but then we also have the Monk scale for when we don't.
Exactly.
And that's not for
Places
regulatory
where it's
submission.
not possible. But
Or for
for
regulatory submission where objective measurements is not feasible.
Exactly, except for that.
Okay, we've got 11 minutes left. We're going to go around. Everybody can state as succinctly as possible what you think should be changed or final comments. Try and keep them very brief. And then we'll wrap up, but we will stick around and have additional discussion for those who want to stick around. Wim, do you want to kick off and
I just said whatever.
You just, would, any comment on ITA?
Yeah, I think the ITA is not the perfect thing, but it's a useful tool right now too, because we agree on it, and I think that we agree on a metric and a color scale is more important than whether it's perfectly linear or whatever. And I think that that gives us a tool that will enable us to compare studies from study to study, and that is very important to get the power as a joint effort to find out whether what is the root cause. And that's I think what I want to see.
Sounds good. Thanks, much, David. Top three.
Good luck. Yeah, I think, probably number one would be decreasing the accuracy limit from 4%. We could agree on that, but at a minimum, 3% minimum. I think having language in there, standardizing the conventional blood reference study right now, with the language of the secondary standard as an option removed at this time, so there's more evidence and the assumptions could be validated, not taking away from all the work that could get to that point. And then a statistically powered increase in sample size, along with diversity built into an increase in the subgroups that we need to define, and how many subgroups, the allocation of samples, allocation of bias, those subgroups, but understanding what we talked about before, separating out, like the Monk scale and including diversity in the trial, then having the secondary piece is the statistically derived amount to be able to detect differences between the groups as we define them.
Okay, thanks, Jake. And Dad.
Yeah, I would I'd reiterate what David said. I'd probably add then to also what Vin was getting at, just some objective measure to really be allow some quantification in what we're trying to do.
Okay, thanks, Mike.
My one big thing that I don't think anybody else will talk about is I want to end up with whatever spec we have ought to be the same for reflectance and a transmissive sensor, because what this is about is talking about what's needed for the patient, not what's easy to do or easier or harder to do for the designer.
Thanks, Mike. Okay.
I'd really underscore the point that was made earlier about safety assessments for all subgroups and equal safety for each group. That study samples should be increased is, I think obvious, but the setting that number, going into this as an outsider, I thought that number would be like in the hundreds to have statistical power to pick up the kinds of errors that Schoeding kind of detected. And it sounds like that's kind of, itself an outlier, but just to put on the table, like how, if these errors impact 1 in 10 or 1 in 20 people, and there are only 5 people of color in a study, that, I think it's an issue. So how to make safety assessments large enough to distinguish patterns from outliers in each subgroup. And then not only about the representation in test groups, but for my kind of third point, to think about that kind of as being inseparable from the diversity of experts we have assembled here. And I think, just thinking forward, this initiative is so important, but there's so much further to go. And about the individualized light measurements, I think calling them objective for now is a bit of a placeholder. As Leo's talk reminded us, it's kind of rounding up. And I think the need for continuing transparency and attention to however that ends up being standardized for now. It would be great to be in dialogue with the National Society for Black Engineers, for example, in the future.
Thanks very much. Leo?
Well, I feel like I've said a lot already, so I don't know if I'm much to add. I didn't get a chance to say about the accuracy question, and I may have missed this part, but sometimes you look at a number and you assume that it's a plus or minus that's insignificant, right? And so if you were to look at a pulse oximeter number and it said 90 plus or minus 5 or 90 plus or minus 1, as a clinician, that might be actionable, right? In terms of how much do I trust this value? Like when we talk about trust in numbers, we look at confidence intervals, we look at plus or minus error. I don't know if there's a way in the pulse oximetry algorithms for that to be included, but if there were a way to do that, I do think that would go some distance towards helping clinicians interpret the information. and knowing when to worry about a number being accurate or when to not worry about a number being accurate. And again, I may have missed, that may have been something people talked about, but if there is an opportunity to do that, that doesn't break the system somehow, I think it would be a powerful addition to how pulse oximetry is interpreted, and potentially even more valuable than knowing the pleth grain, right? So if you know that the device is struggling algorithmically to correct for something, does that increase error or not? I don't know. And I don't know if the hypoxia controlled studies can actually help us determine that. You know, that we see that when we go to lower values, there's a bigger spread and therefore the error is higher. This is outside of my knowledge, but it's like a wish list. If you could add that, I think it would be actionable. Sounds good. Oh.
I think Leo and Carolyn made really great points about skin pigment and the assessment, and I just want to quadruple down on the fact that we really do need to increase the sample size, and I think there needs to be text about the increase of the sample size. I think we can really get away with regulations when you say, oh, only 2 darkly pigmented folks or 15% of the population, when you have a sample size of 20, for example. That means nothing. So I think really having concrete, discrete text about the sample size and improving that and increasing that is paramount. I also, I think objective metrics for skin pigment definitely should be required, and also subjective in terms of what modality is used. That's a little bit over my head, but I definitely think there should be a subjective measure, especially when the reporting of objective metrics necessarily requires some sort of grouping. And so I think some of the subjective modalities can help with the grouping and binning of that.
I just wanted to say that Mike put in the chat a Google Doc that's kind of a straw man that people can please add to and modify. We collected some thoughts about sort of what immediate actions might need to be taken. I want to elevate the excellent points about representation in stakeholder meetings and also in the study population who's at most at risk for having poor health outcomes because of differential bias. And I don't think this is popular. I heard it shot down a couple times, but I would argue that precision in the most clinically sensitive ranges of SpO2 should be a higher value than precision at the extremes that do not require clinical action. Like if you're setting 97 or 100%, like I'm probably not going to do much for you that's different. If you're setting 88% on your SpO2, but I got a blood gas and you're actually like 3%, I might give you COVID therapy that you wouldn't otherwise qualify for. And I think that's what the American population and the press is really picking up on, is differential care. So if we are making clinical decisions based on these numbers, like the question of like how accurate or what is a clinically significant variance, I really think we need to talk about what's acceptable at different ranges, because as a clinician, those are very different questions for me.
But you get your best precision at that high adequate oxygenation. Anyway, I that's a bit of a spanner in the works, but I mean, it's true. You get your highest accuracy, your greatest precision at that range for which you would make no clinical decision. All right, so my top 3 are: let's make sure that the device is appropriately given appropriate attention in the population of intended use. So take, for example, and I'll bring this up, it hasn't been echoed before, but the pinky is of mostly female test subjects is being used as the site for testing neonatal sensor probes. Now, I'll admit it'll fit, but it's absolutely different physio-optics. Blood pressure, fat concentration, the fact that it's going around an ankle of a baby is not represented in an adult female finger. So the same is true of what to do with patients in a home setting. So I would say, since it's opportunistic, you're not getting blood gases, but it's in some settings you might be able to, and Gorham could do that for babies, for example. So I think that's an important situation, is let's make sure that intended use qualifiers is improved within the standard. The second is this issue of signal inadequacy, which right now is undefined. Let's get some common language around what that means, so that, again, manufacturers can meet the bar. And the final one is low perfusion. Right now, that also is not a very defined condition within the standard. And yet we know it's a fulcrum point. We know that you've got crazy low DC signals in that situation, and the pulse oximeters are failing. So anyway, those are my three.
My list is long, but here's the top three. Inclusion of subjects in validation studies reflective of the global demographics sufficient to show lack of bias across groups. Second, indication to the operator of the quality of the saturation estimate. And three, a method of disclosing performance or regulatory status to the user.
That's great. I think we should increase the sample size. I mean, 10's ridiculous. And I think the data shows that somewhere 30, 32 should be a minimum. It's on a well-performing device. If it's a crappy performing device, it's not going to do well in that range anyway. And then I think we need an objective measurement of pigmentation, and that's a half, one of my second ones. And the other half is I think we use the Mach scale for when we can't use the objective. And I think we should change the ARMS and evaluate ways to tighten that up without disallowing all pulse oximeters just because we changed the standard.
Okay, here's an obvious one. Align the FDA and the ISO requirements so there's no carve-outs, like there is right now. You know, it's like, well, we recognize everything except that one clause. Another one was what is the ground truth? Type question like make sure we consider the inaccuracy of the coax when we're doing all this analysis, like what coax, what's inaccuracy. So I would work that into the estimates of whatever inaccuracy spec we use. And the third is kind of one that's not we haven't talked about. Consider the potential differences in some of the descent methods that are used when testing peripheral versus central SpO2 sensors.
Yeah, so I would opt for more inclusive study, like all of us. I think most points are like repeating. One thing is for me really important for the ARMS, because it has been so said here. I would like to stick with the 4% actually to allow for future that we have the chance to have future inventions, you know. So especially because it's over the whole range from 70 to 100. So if you like increase the ARMS, I think that would prevent invention and it would give you a false sense of safety because it's a statistical measurement. And if you say, oh, I have this 1.5%, is that really true, you know, for your whole population? I think it's a false sense of safety you give to a clinician.
Yeah, I would like to write the standard in a way that addresses what I would say the most users. What I'm experiencing is the Woodbine square is not really good understood. If we now add confidence intervals in our specifications, I don't think we as a group probably all understand, but the most technicians that use pulse oximeter readings to do whatever they need to care for the patient, I think it's overwhelming. And that would lead me to my second point is signal inadequacy. Into making a wave interpretation, I think it's also something where you have quite some education. What I am hearing from most customers that are more lay users, I want to have a traffic light. Is it good? Is it marginal? Is it bad? If it's bad, probably we should not show a number anyhow. So I would vote for giving that flexibility to really address the user needs by the manufacturers and not prescribing by the standard how signal inadequacy has to be shown, basically, to allow also improvement on usability over the time and still being compliant to the standard.
Right. So for me, the first priority has got to be protected patients. So it needs to be when somebody uses the product, they need to understand what they're getting into and then be able to make sound decisions off of it. So for me, the target population is going to be people who don't have exposure to measurements, knowledge. That's your CVS, your over-the-counter pulse oximeters, that we protect consumers from using a product that's potentially dangerous for them or for their families, and accept the reality that most pulse oximeters aren't sold to pilots and to super athletes, but they're sold to the COPD or the congestive heart failure patient and those who can't afford. Secondly, I want to level the playing field. If we are creating a standard and putting great thought and insight into it, it should apply equally across all devices, not have two levels of care and just kind of give up an idea that we can afford to sell cheap sensors in the market, even though we know and understand that that's not how they're really being used. We should hold them accountable. And lastly, I agree with we should make sure that we have a mechanism to kind of keep the pulse on the diversity. Diversity is not a static measurement or static thought. So a regular process to make sure that don't get into this mess again. We don't wait too long to update standards to make them representative.
Thanks. First, I'd like to echo what you just said regarding the over-the-counter oximeters. I think that's an area that could be adapted from like the non-invasive blood pressure measurements that one of the gentlemen said online. The second is also chiming in about the increase the number in the bins. And I think that should be led by objective measurements and biostatisticians to determine the statistical power needed in order to accomplish that. The third one is one regarding perfusion. I saw through some of the presentations a confounder of the impacts of these various entities. And if in the course of the clinical validation, we are heating everyone's hands, but then in real life, that's not the case, that may be something that really needs to be at least written about in the document if it's not already.
Thanks, Grant. So my 3 points, greater representation of skin pigmentation, better classification of skin pigmentation, and then tightening up the performance threshold with at least 3% and better ARMS. Sir.
My top 3 would be using an objective skin pigmentation measurement device with the colorimetry technology at a minimum at the measurement site for the pulse oximeter and at probably the forehead to include diverse inclusion criteria for recruitment. And then when that can't be used either for concerned populations, that a well-defined color scale, basically defining all the requirements the Monk scale meets without calling out the Monk scale to allow for other scales to be invented. And then increasing the sample size to something that statistically enables differences to be detected within the subgroups.
Thanks.
Thank you. Increase the number of dark-skinned individuals from the current requirement of 15 or 2. Increase that number to 35, somewhere in that range, or 10. Number 1. Number 2, increase the total sample size to somewhere between 25 and 30, and to make sure that that'll work and get you those numbers. And then number 3, increase the ARMS to less than 2%.
Thanks. John.
I'm a practical guy, so I would love to certainly see a nice distribution, but I want someone who's going to have to give me a statistical test to determine whether my group is outside that distribution. It's very easy to say you want the spread, but I need to know something like that 30% needs to be dark or something like that. That gets challenging. I do not want to see an extreme increase in sample size. We won't be able to get those studies done, and it's just going to favor the larger manufacturers. I think my guess is something like an 18 would be a target, 17 might be good, but I also need to base this on an effect size. These days, in statistics, we're not as concerned with p-values. We want to size the effect. And the size of the effect I'm looking for, and I don't have an absolute parameter, is that the pulse oximeter bias should not create a difference based on skin of more than, let's say, 2% over a saturation range of 80 to 100%. I'm sticking with Caroline's idea that even though I want to include data in the 70% range for the overall AR mass, it doesn't matter as much. So for my effect size, my ideal effect size, I want that over the 80 to 100% range. And it's going to take some work of coming up with an ideal effect size, but that's what we need. Thanks.
Thank you. So I'll start with the ARMS. So when we talk about the current standard, we talk about 3% or 4%, that's only one standard deviation, we all understand. So the errors can be really large. So if a difference between 2% and 3% ARMS can be very huge when you talk about different biases. And it also allows for a different effect size to roll into the spec if you allow for that overall spec to be 3. So first one would be 2% or better. We have seen that most manufacturers are able to do that. So 2% for the RMS. So the second one is to better representation. Like everybody's saying, it should be equal, light and dark. When we have that type spec, if you skew it one versus the other, then you're going to have, representative bias there. So to avoid that, equal distribution is better. And the third one, most critical, also better classification. We all agree, but restating a move away from Fitzpatrick, adapt something that is representative of the entire range. 10 level we talked about among could be messy. We need to validate those scales to maybe a little bit larger extent to say how we can separate the levels within them. But an objective measure would also be good. So far, ITA, based on this group, seems to be also correlating well with 10 level scales.
Sounds good. Thanks for that. From me, I think better communications to the end users, especially at the bedside, about the performance, an objective measure, increased representation, as has been discussed, and then potentially in the standard, calling out that this, because we have limited data, this is going to change, making sure that that is frequent, that that's not, in 10 years, that we acknowledge the limitations of the recommendations that are being made and explicitly either say that we will make a plan or start building that plan for gathering data in a thoughtful way and diversifying how we do that, or diversifying those who are engaged in making those decisions moving forward. Online, encourage everybody to please add into the chat for our panelists who spoke today, just because we're limited in time and can't call on everybody on there, but Ellis, Bumi, Desiree, other speakers, if you want to chime in with your top 3. Sandy, why don't you start with you, and if other speakers and panelists want to chime in, please raise your hand and we'll get to you.
So I'll be pretty quick. I agree much with what you just said, Mike. I think the one piece I would add, or the 1.5 pieces I would add, I think we need to look at what we in the regulatory agency call least burdensome, and we need to find out what the minimum set of evidence is to demonstrate a non-disparate performance. And whether that's just looking at dark and light subjects based on the Monk scale or whatever. That's one thing. And I think the other thing that we need help with is figuring out or developing guides for collecting data appropriately. We can't just say, oh, go out and collect data that's inclusive. We need pretty detailed, explanations on how to do this so that everybody does it consistently and with integrity.
Thanks, Andy. David, you have 30 seconds and we're going to wrap up.
Yep, well, I've said it before. There is a possibility that this effect is the result of variations in the LED frequencies. That should be a fairly easy thing to check, and it should get checked before you embark on the much more expensive suggestions, some of which are very sensible, have come up. But looking at the graphs that we were shown where the ARMS was less than 2%, it showed that the pulse oximeters used in that study were not subject to a bias in the dark-skinned people when they got down to 70%, because they were all within 1% of what they should have done. Thank you.
Thanks, David. All right, well, we've gone 15 minutes over. It's been a very long 2 days. Thanks, everybody, for their attention. We're going to stop the recording. And Desiree, did you want to make a quick comment? And Ellis?
Ellis wrote
Paulus, I see you written a lot. Do you want to share on that?
Sure. I'm just quickly saying that I do think that we do need to increase the representation of darkly pigmented subjects in the studies. I think we should consider oversampling at each end of the skin tone continuum to make sure that we're not missing anyone, that we're fully inclusive. I would also add that even though there may be some hesitancy to move to a new scale after all these years, I think the absence of a standard is also a problem, because at that point you have a vacuum and we don't know what will fill that vacuum. So I do think that it makes sense to say temporarily we will adopt the standard, and I think we should be fully open, and I know I am, and doing more testing on this to potentially update the scale based on evidence, of course, to make sure that the scale is even more inclusive and representative in the future.
Thanks, Ellis. Desiree, did you want to make a comment on pediatric populations?
I just advocate for us not forgetting kids. I know everything always starts in healthy adults and everything like that, but as we see, this does not transfer over to children and size and everything else. And particularly given that like even congenital heart disease is solely based and done off of like pulse oximetry on the right hand and the lower extremity. Things like that need to be heavily considered and taken into consideration as well. So I advocate for including pediatrics into this as a requirement.
Thanks very much. Advocating for kids is a good way to wrap up our 2 days here. Thanks again for everybody's attention. Thank you to all the speakers. Thank you to Fifi and many of the others who made these days possible. Everybody have safe travels, and we'll be in touch soon. You can expect recordings of all the meetings to be online on the Open Oximetry Community Forum within the next several days. We'll also share the meeting minutes. And as a side note, we'll be sending out a few new pieces of data and other things coming down the pipeline, including a new website in the next couple of weeks that will have some more device data and other things. So stay tuned. Thanks for the attention.
Mike, thank you for being master of ceremonies.